Alright guys, welcome back to Underground Science, and in this video we'll be looking at aldosterone and the mid to late distal convoluted tubule in our nephron. Alright, so let's go and get started. So we looked at the loop of Henle in the last video. This video will look at the distal convoluted tubule, in particular the mid to late distal convoluted tubule. So we can zoom into a cell right around there. Alright, but just know in the distal convoluted tubule, we actually can reabsorb some of the sodium, right? Some of the sodium and calcium, and we can actually up the reabsorption of calcium due to parathyroid hormone innervation, right? Through a G-stimulatory pathway in the early distal convoluted tubule, which we'll talk about in a future video. But here, let's look at the mid to late distal convoluted tubule. So let's go ahead and zoom in to a cell right about there of the mid to late distal convoluted tubule. Okay, so let's go and zoom in to this cell right here. So let's say this is our cell. Okay, actually, you know what? That's not good. Sorry about that. Let's go and zoom in, actually draw it in the middle so we have space for certain things. All right, so we have our distal convoluted tubule here, right? And let's say our efferent arteriole in this nephron branches into peritubular capillaries. So that means that we're going to have, so first of all, instead of talking about random stuff, let's go ahead and actually write what this is. So this is it's a zoom in of a mid to late distal convoluted tubule cell. Okay. Okay, now let's say that that's the cell, right? And so the lumen, let's actually show on this side, right? That's the lumen area. And then we'll show a blood capillary, or a peritubular capillary, I should say, a zoomed in version, right about there. All right. So it doesn't really matter what side we put it on. Let's just show it for now. All right. So let's say this side is our lumen, and this side is our peritubular capillary, a branch off of our efferent arteriole. So aldosterone, remember, right, from our zona glomerulosa and aldosterone production is produced, right? Aldosterone is produced, so let's go and write this down. Aldosterone is actually produced in our zona glomerulosa of our what? Adrenal cortex, right? Adrenal, it's our first layer of our adrenal cortex. And remember, aldosterone is not a protein hormone, it's a steroid hormone. So it's a steroid hormone derived from cholesterol. So it's a steroid hormone. All right. And when we produce aldosterone, we said that it can go into the mid to late distal convoluted tubule cell and increase sodium and water reabsorption into the blood. Now let's see how. All right, so remember, aldosterone is a steroid hormone, so it's not going to come here and bind because it's not a peptide hormone. It's actually going to cross the phospholipid bilayer, and it's going to bind to a transcription factor. So let's say this is our nucleus, and we have our DNA here. Okay, so we have our DNA here, and it's going to bind to a transcription factor inside the cell. So let's say we have a transcription factor, right, that's inside the cell, and this guy's just waiting, right, so for something to bind. Okay, and so let's say we have aldosterone that comes here and binds to this transcription factor. All right, and once aldosterone, so let's indicate aldosterone by this green dot, once it binds to this transcription factor gets activated and actually goes and binds to a segment of the DNA, where if we transcribe those genes and translate those genes, right? So if we undergo transcription into mRNA, then translation into a ribosome, and then Golgi, apparatus, and ER, and finally get our proteins, what's going to happen is that these proteins that we're going to make through transcription and translation are actually going to be sodium-potassium pumps, sodium-potassium ATPase pumps. So through our transcription and translation process, through protein synthesis, it's going to result in many of these sodium, right, potassium, ATPase pumps. And these sodium, potassium, ATPase pumps are going to be put on the basolateral side of our mid to late distal convoluted tubule cell, so near the blood. near the peritubular capillaries. All right, and the reason being is because remember, aldosterone secretion, guys, was triggered by low sodium levels in the blood and high potassium levels in the blood, right? Those were the two main triggers. So let's go and write that down. So it's triggered by low sodium levels in the blood and high potassium levels in the blood. Remember, low sodium levels in the blood, we'll talk about, are really activated the macula densa cells in the distal convoluted tubule of the kidney, right? And then it further activated the juxtaglomerular cells in the afferent arteriole, all right? And high potassium levels can also trigger the adrenal cortex to make aldosterone, all right? So to lower the potassium levels and increase the sodium levels, that's what aldosterone needs to do. And so that's why we put sodium potassium ATPase pumps through transcription translation, right? Because aldosterone activates a transcription factor, and these sodium potassium ATPase pumps cause 3 sodium to leave into the blood, and then 2 sodiums from the blood to come in, right? And this process uses ATP, and this happens in all of these. So 3 sodium cations will leave, and 2 potassium cations will come in. So see what we're doing here, we're increasing blood sodium levels, and we're also decreasing blood potassium levels, which is what the initial trigger was, right, for aldosterone secretion. And what happens here now, so we just solved this problem, right? But what happens is that we need sodium to come into the distal convoluted tubule cell, right? And the mid to late distal convoluted tubule cell, and we need potassium to leave. So what happens is that this transcription factor also activates certain genes to transcribe and translate into proteins, and these proteins are going to be sodium channel proteins and potassium channel proteins. So we're going to have sodium channel proteins, all right, so let's draw 2 here, and we're going to have potassium channel proteins. Let's draw those, and we're going to have potassium channel proteins in purple. The reason why we need sodium to come in from the lumen, crap, we didn't label that, from the lumen, right? So the reason why we need sodium to come from the lumen into the mid to late distal convoluted tubule cell is because, see here, sodium is constantly leaving the cell, right? From low to high concentration. So we need to replenish the sodium concentration here. And so what's going to happen is that sodium from high concentration over here in the lumen is going to follow into the cell where there's low concentration of sodium. All right, so let's go and draw this here too. And then these sodiums that come in can participate in these sodium potassium ATPase pumps. Now potassium is going from low concentration, which is in the blood. Okay, so this is the blood peritubular capillary. So potassium is going from low to high concentration. That's why to maintain this low to high concentration gradient, we have to have potassium leave the cell, right? It's going to automatically want to leave the cell is what I'm trying to say, because usually, right, molecules want to go from high to low concentration. Potassium is high concentration inside. So potassium would naturally want to go from high to low concentration. All right, so potassium would naturally want to go from high to low concentration. So this is where there's low concentration, this is where it's high concentration. All right, so let's draw the same thing here. All right, so it's where there's low concentration, where there's potassium. And that's pretty much it. So we end up reabsorbing sodium. Oh, and also in the mid to late distal convoluted tubule cell, along with sodium, water also follows sodium to be reabsorbed. So we also have water that follows the sodium, right? Because water follows osmosis from high to low concentration, right? It follows the solutes, and water also gets reabsorbed into the blood as it follows sodium. Now, the main triggers, right, for aldosterone secretion was low sodium, high potassium, and the reason why we initially needed to secrete aldosterone was because the blood pressure and blood volume was low, right? So what happened was, now that we increased sodium in the blood, we fixed this problem. We decreased potassium in the blood, we fixed this problem. And because we also reabsorbed water into the blood, we increase the blood volume, thereby increasing the total cardiac fluid return, right? So that we increase preload, and then that led to the increase in blood pressure. So we fixed this problem also. All right, and it turns out that it's only wherever aldosterone works is where water is permeable. After that, when we get to the collecting duct with the principal cells and the very late distal convoluted tubule, then water is not able to cross into the cell and into the blood. We need hormones like antidiuretic hormone for that to happen, and we'll see that in we'll take a look about what happens when antidiuretic hormone acts on the principal cells of the collecting duct, all right, in a future video. But this is all I wanted to get through in this video about how the basic idea of how aldosterone acts on the mid to late distal convoluted tubule cells, and how we actually fix the problems about increasing blood pressure, increasing blood volume, decreasing blood potassium, and increasing blood sodium levels. All right, and how aldosterone cross directly activate transcription factors to transcribe and translate proteins such as sodium potassium ATPase pumps and sodium and potassium channels, all right, on the apical side of the cell. Okay, so I hope this video made sense to you guys. I hope to see you guys in my future videos. We'll talk about more cool topics about biochem, physics, and chem. Don't forget to hit that notification bell, like, and subscribe. See you guys later, and stay safe.